Okay, so today we're talking about genetically modified organisms and how we can have these. So we usually think about things being genetically modified like corn to be disease, drought, or pest resistance, or even to increase nutrition. But we can also genetically modified animals for research, or even mosquitoes to fight disease. But a big thing that we don't maybe always think about is that we can genetically modified organisms in order to develop drugs like insulin, hormones, vaccines, and anti-cancer drugs are all from genetically modified organisms. And this is what our lab was dealing with today. Okay, so we can think about this with the context of insulin. How is insulin made? So in 1922, you don't have to memorize any of this. I'm just giving you some context for an example. But Canadian researchers isolated insulin and it cures diabetes, and it was bovine insulin, so from a cow. And they were able to win the Nobel Prize in 1923. And previously, diabetes was basically a death sentence. So there was no treatment, no cure for that disease. And then now, in 1978, and this is what we're going to use now, is a way to produce human insulin much faster than extracting it from cows, and that is from the use of generating and genetically engineering bacteria, and specifically E. coli, to have recombinant DNA. So we are recombining the DNA to have the instructions that we want it to make, in this case, insulin. And this was approved by the FDA in 1982, and that is what is currently used. So you can see here just some numbers to give you the price, so that if we have things that are, if we have insulin made with E. coli, it's going to cost about $60 per gram. But there are other things for other disorders like human growth hormone that's going to be more expensive. And all of these are cheaper than gold. So it still is expensive. Okay, how this works though is the idea is that first we need to find the gene for the protein that we want. In this case, we were talking about insulin, but we have the gene, we find the sequence. Okay, then we take that gene and we are going to put it into a bacteria, and we are going to use plasmids, those circular little pieces of DNA that are it will contain our gene, and we want to put it inside of our bacteria. So then we're going to grow lots of that bacteria that now has our gene inside of it, and inside of it, it's going to be taking that gene, turning it to mRNA, making a protein, so going through transcription and translation to make our final project. So basically like little mini protein factories, which is super great and helpful. Then we can purify out and extract, we can purify out just that protein that we want. Okay, so how this happens is, again, I mentioned, is our plasmids, and that plasmid contains the sequence of our insulin or whatever thing we are wanting to be expressed in the E. coli. Okay, so we have to first make our plasmid, then we do a bacterial transformation, okay, and that's what we did today, and that is taking the plasmid and putting it inside to the E. coli. And this is done by heat shocking, so basically you take the E. coli and you put it in really hot water and then in cold ice, it's not very long, ice for 15 minutes, heat shock for 50 seconds, and then ice for 2 minutes, and then we put it on our plates and let them grow. And so we're able to look at what is produced. Okay, so we have our plasmids and they can make our proteins. Okay, so yep, these are circular pieces of DNA. Okay, so some other things that are located on our plasmids are a sequence that has something that's going to code for some sort of protein that's going to allow the bacteria with this particular plasmid to have antibiotic resistance, because we do not have 100% of the E. coli will not get our plasmid inside of it. It's just not a very efficient process, but that's okay. We don't need it to be efficient. We just need some of them to get the correct gene in it, but we need to be able to identify it. So we add an extra gene, and that is for an antibiotic resistant in a protein that allows for the E. coli to become antibiotic resistant. And that is how we are able to select for only the E. coli that contain our plasmid inside of it. Okay, so the protein that we are using in our experiment is for green fluorescent protein. So instead of adding the sequence for insulin and making insulin, our E. coli are going to make green fluorescent protein. And this is helpful because we're going to be able to visualize it and see that it is in fact making the protein we want, because it is a protein from jellyfish and it fluoresces under UV light, as its name suggests. Okay, so here is the specific plasmid that we are using. It's going to have our green fluorescent protein. It's going to have an ampicillin-resistant gene that allows for the bacteria to survive on ampicillin, so that we can make sure that only the bacteria with our plasmid are there. But we also have a regulatory protein that is going to be involved too. And we've talked about this with our gene expression unit, that regulatory proteins can switch on or off gene expression. So in this case, we have a regulatory gene as well. And that is for ARA-C. And so that means if we grow, this is only going to be turned on in the presence of ARA. Okay, so just a reminder that our DNA with our plasmid here, what happens when we transform our, so we have our E. coli, we're going to add our plasmid. When we transform the plasmid, our DNA is negative. We use this characteristic of DNA also in that gel electrophoresis experiment. But we're going to, one of the solutions we add into our transformation is calcium ions, and it's basically going to help neutralize our charge on our DNA so that it's able to slip through that cell membrane easier, because remember, we have a hydrophobic center of our membrane. Okay, then we heat it up, and what that does is basically makes little pores inside of the cell membrane. We don't exactly know why it does this, but it is a phenomenon that we exploit. So we heat it up, cool it down, and it allows that plasmid to enter into the membrane. Then the pores close behind it when we cool it down. Okay, then we're going to add broth to it. We call it LB. I'm not sure, I don't remember off the top of my head what LB stands for. I think it's just a particular type of broth. It's basically got all the extra things that our E. coli need to grow and express our protein. Okay, so here's an example where we have our E. coli. We're adding that plasmid to only some of them, and then they're able to now make our ampicillin-resistant protein. That means when we have ampicillin present, that the cells that do not have our plasmid are not going to be able to survive, and the only ones remaining are the ones that contain our plasmid, which is what we want. Okay, we grow it in this broth. It's basically kind of like a soup broth, but it's got all the things that it needs to grow: salts, vitamins, nucleic acid, amino acids, nucleotides, carbohydrates, everything it needs. Okay, and these are kind of the steps in the summary. And this is what we did in class. I'm going to kind of skip through this. We're going to put our DNA or our E. coli into our tubes. We add our, we either add or don't for our control the plasmid. We put it on ice. We're going to heat shock it. And then we put it onto our different plates. Here's the heat shock. Okay, and then we have that regulatory protein that is going to be there. So again, it's going to, arabinose is a sugar, and it's going to work like a switch. It's going to regulate. So without arabinose, the switch is off, which means it's going to be blocked by, it's going to block RNA polymerase to come and transcribe our gene. So that means if we add arabinose, then it's going to remove this negative regulator, and it's going to be able to express our gene. And then they'll be green.